In this video we will discuss about immunotherapy and go through a brief example. An example in brief about how this immunotherapy has been implemented, or people are trying to implement this immunotherapy. So the immunotherapy is about helping the immune cell to learn and detect cancer or tumor cells and kill them. As we have discussed in previous videos that the slow response or the weak response of immune cells might lead to cancers, or might lead to uncontrolled growth of cancer, or the growth of cancer to that extent that it is difficult to cure or with at present situation. So where are most of the immune cells lies? Most of the immune cells lies in white blood cell, and that too a specific kind of white blood cells that are called lymphocytes. They are majorly categorized into three types. One is B lymphocyte, T lymphocyte, and natural killer cells. B lymphocytes function is to identify the uh, foreign particles or tumor and kill them, and T lymphocytes has similar function, but apart from that it also helps the B cells to recognize and kill the tumors. And natural killer cells works independently, uh, probably we can say that works independently from T and B cells, and it also identify the infected cells and kill them. So now let us understand this immunotherapy with the help of an example, that is chimeric antigen receptor T cell therapy. It is from the name itself, it is suggested that it is related to T cell therapy. So when a patient who is infected with a cancer, his blood has been circulated through a machine from which T cells are being extracted and blood are being recirculated back into the patient, and this T cells then goes to a lab or manufacturing facilities where with the help of infected virus a gene is transfected, or you can say gene is integrated into the host cell genome, and this can be done this is being done with the help of stable transfection, and we have discussed what is stable transfection in our previous videos. So here they have used inactive virus to transfect the gene into the T cells, gene of interest. So what this gene of interest does is the gene of interest leads to the specific kind of receptor at the surface of the cells or at the plasma membrane, uh, outside the plasma membrane, and that can recognize that can recognize the antigen on the cancer cells. So these specific kind of receptor are called chimeric antigen receptor, or CAR. And when this T cells, then the T cells are continues to grow in vitro to an extent that it is, population is enough for transferring these cells into the patient's body. So this is usually around millions of attacker cells, and these millions of attacker cells are put back into the patient, and these receptors are attracted towards the antigen, or we can say antigen that is present on the surface of the cancer cells. As we can see in the figure, these modified T cells can recognize the antigen that is specifically present on cancer cell and then kill them. After that, as we know that stable transfection remain for a longer duration of time. So this CAR T cell will remain for a longer duration of time to prevent the returning of the cancer cells. So in this way, the immunotherapy, or we can say more specifically chimeric antigen receptor T cell therapy works. So this is the basic idea behind this chimeric antigen T cell receptor. And one more thing we need to understand in this is that while transferring these cells back to the patient, we need to do the most appropriate time is to do just after chemotherapy, because after chemotherapy, as we understand, chemotherapy based on DNA damage, we can understand that at that point of time the amount of tumors are very less, so they don't have to do too much function at that time. At the same time, uh, the efficiency of this T cells will be good, because of the less tumors present in the patient at that time, so that it will get time to adjust to the patient's body. At the same time, uh, their efficiency is more compared to the other situation when these cells are being in transferred to the patient. So as it is written also here, that typically these T cells are transferred after chemotherapy is given to make space and continue to multiply. So that's all for this video. Thank you for your time.